We've already established that the cell membrane is selectively permeable. Some things move across the membrane, some things have to have help to move across the membrane, and some things just don't move across the membrane because remember, we want to regulate the intracellular fluid, the extracellular fluid, what gets in, what gets out of the cell. So before we can do that, we need to understand what gradient refers to. So we have gradients across the cell membrane. Gradient simply means there's more of something in one place as compared to another. The simplest is the concentration gradient. So if you look at the image on the left side of the screen, we have a container that has a semi-permeable membrane running right down the center, which means some things can cross and some things can't. Now notice on this side, we have more little blue dots. On this side, we have fewer little blue dots. So in this solution, the little blue dots are the solute, whatever that chemical is. And because there's more on one side of the membrane than the other, then there is a concentration gradient. Concentration just means how much of something is there in something else. Okay? Now, an electrical gradient, same idea, but we're not talking about the chemicals, we're talking about the charges. So if you look at the image on the right side of the screen, you see on this, we have more positive charges on this side of the membrane. We have more negative charges on this side of the membrane. So that means we have an electrical gradient. So now keep in mind, this is just representing inside the cell versus outside the cell. Inside the cell versus outside the cell. An electrochemical gradient is a combination of the two. So there's a different concentration of the chemicals and there's different concentration of the electrical. So different gradients rather than concentration. Okay? So just keep in mind it means there's more of something on one side of the membrane versus the other. When ions, molecules, water move into or out of the cell, that is called membrane transport. And we break that into two major categories based on whether or not it requires energy. Now remember, our energy is ATP, right? We have to use that fuel for the energy to do work, right? Okay, so we break it into two categories. Passive transport or passive processes do not require any energy to be input for the action to occur. So whatever it is can cross the membrane without having to spend any ATP. So it relies on the kinetic energy of random movement of molecules. Now, we have two different types of passive processes to move things across the membrane, and that is diffusion and osmosis. So we're going to talk about each one of these individually. But the big idea here is represented by this image very easily. If you've got a man standing at the top of the hill, he can push the rock and it'll just go down the hill. Or if the rock is sitting on the edge, it's just going to fall down the hill, right? Now, active process, we've got to put energy in. So active processes would be, okay, this poor little man now has got to start at the bottom of the hill and push the rock all the way up. No energy needed for down the hill. Lots of energy needed to go up the hill, okay? So the other key thing to remember here is with a passive process, molecules are moving from where there's more to where there's less to try to reach an equilibrium. So let's go back to that previous slide for a moment. So let's look right here. We've got more blue dots on the left side versus the right side. So let's say this is intracellular fluid. This is extracellular fluid. So we've got more blue dots inside the cell than outside the cell. Blue dots will move across the semi-permeable membrane as long as it is permeable to little blue dots. And these little blue dots will continue to move until we've reached an equal concentration of blue dots on each side of the membrane. Okay? So we move things from where there's more to where there's less. That doesn't require any energy. It just happens. Active process, not only does it require ATP, but one of the main reasons it requires ATP is because we're pushing the rock uphill. So we're moving things from where there's already less to where there's more. Let's go back to that other slide for just a moment. Okay, so let's stay right here with the same image. So we've got more blue dots on the left, fewer blue dots on the right. So there's more on this side in the intracellular fluid than there is the extracellular fluid. So normally these would just move to reach equilibrium. That's just going to happen with no energy. But if we want to take the blue dots in the extracellular fluid and move them where there's already more, that requires work. So think of that as we're pushing the rock uphill, because it's not just going to happen trying to reach equilibrium. We're making it more different instead of closer to the same. So that is the energy ATP needed to cause it to move from low concentration to high. We have two types of that as well, active transport and vesicular transport, that we're going to examine these processes on the next couple of slides. So this is a flow chart that just gives you a summary of the active and passive processes of membrane transport. What I recommend is don't worry about this one yet. Get to the end of this discussion, then come back and walk yourself through this flow chart, and you'll see how simple this is to understand. The first type of cell movement we're going to look at, or membrane transport, is a passive action called diffusion. So diffusion is the movement of ions or molecules down their concentration gradient, which means from where there's more to where there's less. Diffusion happens because of the kinetic energy of the ions or the molecules. So kinetic energy is random. It's a constant motion of ions and molecules. So when they bump into each other, they bounce in different directions. And this causes the molecules to move throughout that solution, or to spread out and diffuse, hence the name diffusion. So again, we're moving substances from where there's more to where there's less. There's several things that can affect how quickly and how well that happens. The warmer the temperature and the steeper the concentration gradient, the more quickly diffusion occurs. Excuse me. Now, this happens if the molecules are unopposed, so there's nothing stopping diffusion from occurring. Ultimately, the movement of all of these substances will allow that molecule or that ion, whatever it is, to reach what we call equilibrium, which means everything is the same. You've probably done this before if you've ever dyed Easter eggs. Let's look at the top image. You start out by putting your dye pellet in a container of water. Well, there's a very steep gradient there because all of the dye is in the pellet. So it's going to start very quickly diffusing out through the water. And as long as it sits there and you don't do anything to disturb it or stop it from happening, it will continue to diffuse. The dye particles will diffuse throughout the water until it has reached equilibrium, meaning it's evenly distributed throughout. Now, the same thing happens with ions, with molecules moving across the cell membrane. They move until they reach equilibrium. There are two different types of diffusion. We'll start with simple diffusion, and this is when the molecules move through the membrane unassisted. Now notice that's underlined. That should tell you the next slide's going to be something different, right? So these unassisted molecules are not regulated by the cell membrane or the plasma membrane. They will just continue to move across that membrane as long as there is a concentration gradient. Again, that simply means they'll keep moving as long as there's more inside the cell versus outside, or outside versus inside. This happens with small nonpolar lipid-soluble molecules. It's non-specific, so we don't have to have a specific protein to allow it to get in. It's a slow process, and it slows even more as it reaches closer and closer to equilibrium. This is how our respiratory gases, such as oxygen and CO2, some of the fatty acids, ethanol, urea, alcohol, and fat-soluble vitamins move across the cell membrane. To facilitate means to help, to help or assist something to happen. So small molecules that are charged or polar, so charged ions, charged molecules, polar molecules, they're basically blocked from getting across the plasma membrane. So they have to be transported in or out of the membrane using a protein in the membrane. The protein facilitates the act of diffusion. Now, these proteins are very specific, so they only allow one type of molecule or ion to cross, and that is the only thing that can use that protein. So some examples of this are glucose, ions, and amino acids. Now, there are two types of facilitated diffusion. Channel-mediated diffusion is basically a leak channel, where things will just leak through that opening in the protein from where there's more to where there's less. It's still diffusion. Things are moving from where there's more to where there's less. So channel mediated, the channel mediates or helps it to happen. Okay? Your carrier mediated is also very specific, also moving things from where there's more to where there's less. Carriers only bind to one type of molecule or ion. When the blue dots bind to the protein changes shape, opens up, and whatever the blue dot is now crosses the cell membrane. And it cannot get across without that carrier facilitating the movement. Just to recap this channel-mediated diffusion, small ions are going to move through these protein channels, one type of ion per protein. So per channel, only one thing will fit through. So your leak channels are always open. So there's nothing covering them, nothing stopping the ions from moving from outside to in or inside to out. Gated channels, they sort of think of it like a gate. You have to open the gate to get through. So they're usually closed, but they open in response to whatever that ion is that is trying to get through the gate and cross the membrane. Carrier mediated, remember, has to have some help. So small polar molecules use a carrier protein to get from one side of the cell membrane to the other. The binding causes the carrier to change shapes, and when it does so, it opens up and releases whatever that solute is to the other side of the membrane. A uniporter is one of these transport proteins that moves one solute. Uni means one. So the number of channels and the number of carriers is going to determine how quickly those solutes can cross the cell membrane. So what happens if the solutes can't cross the cell membrane, if they're non-permeable, such as your charged or polar or big solutes? So some ions and glucose and protein, if they can't move across the cell membrane, what's going to happen? Our body still wants equilibrium. So let's look up here at this image. So on the far left side, we've got the red dotted line representing a semi-permeable membrane. Let's say that the left side of this container is extracellular fluid. The right side is intracellular fluid. Now notice the total volume is exactly the same, right? Now I'm talking total volume. But which side, inside the cell or outside the cell, has more solute? Well, it should be obvious. This one right here, outside the cell, has more solute. So that means this side of the cell, or this side of the cell membrane, has more water. Yeah, well, that's pretty self-explanatory. If the volume is exactly the same and one has more solutes, then the other has more water. So we call this difference, of course, as we've seen already, concentration. So water will move if the solutes cannot, because our body still wants to achieve equilibrium. For example, water moves down its concentration gradient if there's a solution that is 1%. Okay, now let me back up. Strike that and let me start again. Okay, so let's say on one side of a cell membrane we have a 1% solution. That means 1% is solutes, 99% is water. On the other side, we have a cell that has a 3% solution. That means there's 97% water, 3% solutes. So there's a concentration gradient. One side has more water than the other, okay? The other side has more solutes than the first one. So water will move if the solutes can't until equilibrium is reached. Water is constantly moving across cell membranes. It can just slip between the molecules in the phospholipid bilayer, or it can move through aquaporins. Let's look at that word. Aqua means water. I knew you knew that. And pore is a hole. So water hole. Now, that water's going to move back and forth, and it will just continue to do so. But when you've got a different concentration on each side of the membrane, the water's going to move to try to get to equilibrium, not just passively flowing in and out and not making any changes. So right here, remember, we've got more solutes on this side than this side. So water would move from the side where there is more water to the side where there is less water. And then what happens? The volume goes up on this side, volume goes down on this side, but the concentration has reached equilibrium. Before I move on to the next slide, I want to point out something about this little cartoon I added here at the bottom. So you've got 4 gentlemen in a boat, and the boat is on the water, and obviously there's a hole in the boat because water is moving from where there's more in the ocean to where there's less in the boat. That's a passive process, okay? That's like osmosis. But what are these 2 dudes doing? They're bailing water out. So they're trying to move water from where there's less inside the boat to where there's more back out in the ocean. That's a passive process. Is it requiring energy? Heck yeah, it is. Those 2 dudes are burning energy. So in that one image, we've got an example of passive and active movement of water. So the concentration gradient between the cytosol, the fluid inside the cell, and the extracellular fluid or interstitial tissue fluid is going to create a difference when there's more water on one side than the other, more solutes on the other side versus the one, right? So if we look at this, the little water molecules here, there's your little red oxygens and your little 2 hydrogens attached. So look, there's a whole lot more water molecules in the extracellular fluid than there is inside the cell in the intracellular fluid. So water will just passively diffuse across the membrane or go through the aquaporin, the channel. Now that's going to continue to happen until we reach equilibrium, which means equal concentration on both sides of the membrane. So here inside the cell, we have a higher solute concentration. That means there's less water. Outside the cell, we have a higher water concentration, so that means there's less solutes. All right, for you guys in my online class, I don't get to see you face to face for lecture. So you're going to learn a lot about me and the way my brain works by listening to these lectures. So I've been sitting here for a little while looking at this picture, and I've decided I really don't like it. So I'm going to use the information on this slide, but I'm going to go back to an image we've seen previously. So let's back up right here and use this image. So osmolarity simply refers to how much stuff is in a solution, okay? How much What is the concentration of solutes? So let's go back right here on the left side of the picture again, okay? So we have just a few solutes on this side. We'll call this side A. And we have more solutes over here on this side. We'll call it side B. Let me see if I can get my stylus to work. A and B. Okay. So A has more water. B has more solutes. So side B is going to try to take water from side A. So I say that side B is sucking water from side A. Okay? Now side B has what we call a higher osmotic pressure. The simple way to understand that is the more solutes in a solution, the higher the osmotic pressure. So I say osmotic pressure sucks, because we just said that side B was going to suck water from side A. So the higher the osmotic pressure, the more water will be sucked from the other side to try to reach equilibrium. Now let's go back to side A again. Side A has more water. Hydro refers to water, so side A has a higher hydrostatic pressure. Now what is that going to do? The water from side A is going to push through the membrane to side B. So hydrostatic pressure pushes, osmotic pressure sucks. But they're both moving water from side A to side B until we reach equal concentration on both sides of the membrane. Okay, I hope that made sense to you. We're going to talk about this a little bit more, so let's continue. Now, why do you think all of this is important? Well, we've got a lot of water in our body, right? So cells are constantly gaining and losing water through osmosis. Water moves between the cytosol inside the cell to the extracellular fluid. So we're constantly just moving fluids around. Well, that can affect our cells. So tonicity refers to how well the cell can maintain its shape during all of this water moving in and out across the cell membrane. So there are three types of tonicity. Iso means same, so same concentration. Hypertonic, hyper means above, over, too much. So a hypertonic concentration has more solutes than another. A hypotonic, hypo means low. So a hypotonic solution has fewer solutes than another. Let's go back and look at that picture again. Okay? Side A is hypotonic to side B because it has fewer solutes. Side B is hypertonic to side A because it has more solutes. All right, keep that in mind. We're going to keep talking about this. Now, as I'm going through this, I want you to think about why I put this picture here of this healthcare provider hanging an IV bag. Okay, iso, remember, means same. So the fluid within the cell and the fluid outside of the cell have the same concentration. So inside the cell, in normal erythrocytes, red blood cells, normal plasma, we have 0.9% saline solution outside of the cell and 0.9 saline solution inside the cell. So the fluids are isotonic to each other. Now, normal saline that we have in IV fluids is 0.9% saline. Now let's go right back up here. If we've got 0.9% inside, 0.9% outside, we're already at equilibrium. That doesn't mean water is not going to move, because we've already said water is always moving across the cell membrane. It simply means water moves in, water moves out, water moves in, water moves out. The cell doesn't change. So no net movement of fluids in an isotonic solution. Hypotonic, remember, hypo means under. So outside, the extracellular fluid, also called the interstitial fluid, has less solutes than inside the cell fluids, more solutes. Okay? So that means outside the cell is hypotonic to inside the cell. Now, we've already established water moves from where there's more to where there's less. So the water's going to move from outside here in the interstitial fluid into the cell. That increases the volume of fluid in the cell, increases the pressure on the cell membrane, and can cause the cell to lyse or to burst, sort of like overfilling a water balloon. That's called hemolysis. Well, when hemolysis occurs, that cell can't do its job, and its job is to carry oxygen out to your body tissues, so you have a decreased oxygen carrying capacity. And then we have the opposite condition. Remember, hyper means too much, above, over. So when we have a hypertonic extracellular fluid, that means there are more solutes outside the cell, fewer solutes inside the cell, so there's more water now by concentration inside the cell than outside. Water moves from where there's more water to where there's less, so the cell is losing water and it's dehydrating. We call that crenation, C-R-E-N-A-T-I-O-N. So the action is cells will crenate. Again, what happens? Well, the erythrocytes, this right here says hypertonic. So let's back up. Draw a line through that because that's not what we want, guys. Crenation occurs, not hemolysis. Okay? Let's just do that. So when the cell loses its water, if it doesn't regain the water pretty quickly, that cell is dehydrated and also cannot do its job, which is to carry oxygen.